Hi, welcome to our panel discussion where we're going to be talking about some of the recent trends in screening G protein coupled receptors, GPCRs. And I really am excited about the people that are going to be speaking on this panel. We make this a really engaging and informative time over the next hour. I'm going to start with just some introductions. I'm going to let each of the panelists actually introduce themselves. I'm the moderator. I'm Annette Gilchrist. I'm an associate professor at Midwestern University, which is just outside Chicago, where currently it is freezing cold. I have a second company that I recently started called My Genome RX, which has to do with pharmacogenomics. And I worked in the field of GPCRs for a couple decades. I did a postdoc with Heidi Ham, and together we started a company based on the interactions between G protein coupled receptors and G proteins. And so I have, for many years, studied that receptor G protein interaction and how that really shapes how the cells signal and how you can screen for them. I'm a big fan of functional selectivity and allosteric modulators, which I think we'll touch on today. And so I'm really happy that I have the opportunity to introduce some of the panelists that are going to be talking today. So I'm actually going to ask Kevin to start and introduce yourself and tell us a little bit about your life in the world of GPCRs.
Thanks very much, Annette. It's wonderful to be here and welcome everybody. I'm Kevin Pfleger. I'm a professor at the University of Western Australia and the Harry Perkins Institute of Medical Research. I'm head of molecular endocrinology and pharmacology there and have been for many years now. And I'm also director of biomedical innovation, so I have a broader portfolio of responsibility there as well. And I'm also chief scientific advisor of Dimerix Limited, which is a spin-out company which is looking particularly at receptor complexes and heteromers and trying to understand the novel pharmacology of those.
Juan, how about you next?
Thank you, Annette. So my name is Juan José Fung. I am currently principal scientist at GPCR Therapeutics, a drug discovery company focused on targeting GPCR heteromers in cancer. The company headquarters is in Seoul, Korea, but we have a small group here in the San Francisco Bay Area. I received my academic training in the lab of Dr. Brian Kobilka at Stanford University, studying GPCR dimers and structural dynamics. I've spent significant time in industry studying membrane proteins and performing assay development and high-throughput screening. And currently, I am focused on assay development and new technologies to bridge the gap between in vitro and in vivo GPCR heteromer pharmacology.
Yamina.
Hi everyone, so I'm Yamina Berchiche. I have a little bit different background, or at least different occupation right now. I'm the founder of Dr. GPCR, which is an ecosystem designed to bring together stakeholders interested in understanding GPCRs, G protein coupled receptors, which, as we all know, control virtually everything in the body. And the mission of Dr. GPCR is to bring us all together, all of us who love GPCRs, whether we're interested in dimers, heterodimers, homodimers, or even as functional selectivity, so that we can work together to better understand and better target the receptors. And within the context of Dr. GPCR, I have created different programs. One of them, and thank you, Annette, for inviting me, is the Dr. GPCR podcast. And I've had two of you here on the podcast, Kevin and Annette. And the idea is to promote and talk about all these wonderful scientists who spend decades of their life working on GPCRs. Outside of Dr. GPCR, I have my training in biochemistry, but I've worked in the field for about 20 years. I worked on chemokine receptors, and I've used different biophysical methods to better understand structure-function relationships of chemokine receptors, including functional selectivity. And I'm really excited to be here today. Thank you.
Aarti.
Hi, my name is Aarti Kawatkar. I work in AstraZeneca, Boston, and unlike you all, I'm trained as an organic chemist, and as a profession, I moved few years before to chemical biology group in AstraZeneca. So I started as a medicinal chemist. I work on different small molecules, and then when in AstraZeneca we opened chemical biology group, I moved there, and I'm part of target deconvolation and target engagement team. So my main focus is working on small molecule target deconvolation and target engagement, and I support multiple drug discovery projects at different stages, from hit identification to lead optimization, even the clinical candidate selection. So unlike you all, I won't say that I'm expert in GPCR, but I work on many different type of proteins, depends on the soluble protein, then membrane protein, single pass, multipass, and then I have worked on even GPCR proteins. And recently, we have published a paper on cellular thermal shift assay, CETSA, which can be used as one of the screening methods for GPCR proteins.
I'm sure we'll talk about that later, that paper. That was great. Ajay, do you mind?
Sure, absolutely. Thank you, first of all, for having me over, and hello, everybody. It's really nice to kind of be with all of you today. I'm Ajay Yekkirala. I'm co-founder and chief scientific officer of Blue Therapeutics. We are a platform company built around receptor interactomics. So we target GPCR heteromers, non-GPCR ones. So there's so many receptors that dimerize for functionality. So we target all of them, and our core areas are in CNS and oncology, but we're also expanding into, infectious disease and so on. My academic background is a little bit varied as well. So I was in a medicinal chemistry background initially with Phil Portuguese at University of Minnesota. We were targeting making ligands for opioid, mGluR5, cannabinoid, all kinds of receptor heteromers. Then I moved on to Harvard, where I was with Clifford Wolf. We worked on nerve regeneration, spinal cord injury, and also neurobiology of pain. Before starting the company. So yeah, I'm super excited. We can talk about dimers, GPCRs, whatever have you. Exciting.
Well, I'm actually excited to talk about dimers, and let's start there, or heteromers, as the case may be, right, Kevin? You'll correct me, I'm sure. So, it's kind of funny because, it really was only 10 years ago, I think, that I sat in a meeting and listened to a debate about whether dimers were in fact real or not. It's really not been that long ago. Certainly within my career in GPCRs, the argument about whether dimers actually form and if they're physiologically relevant. But I really think that's pretty much been put to bed. But I think we still wonder about how do you target them, why do they form, what differences are there, do the drugs that we already have bind to the dimers or not? And so maybe you guys can talk about this a bit in terms of when you target a receptor, do you think about, does it form a dimer? Does it matter that it forms a dimer? Do you wonder, what it forms a dimer with and whether it matters?
Yeah, it's great questions, and I've again been through the field for many years and experienced that change. I think it's incredibly important to think about complexes of proteins and how, if you think of a GPCR sitting in a membrane, it's not like to be sitting there on its own. In fact, it won't be. It needs to signal. It needs to interact with other proteins, and the complex it forms will determine its pharmacology. We have different allosteric modulations happening between proteins, and if those interactions affect the conformation and affect the ability of the ligand to bind, to interact with its signaling molecules and downstream effectors, it will change the pharmacology. So understanding the importance of that is critical. The question is, where does that occur? When? In which cellular context? And really understanding the relevance of the pharmacology. And that's really why these amazing technologies have developed over the years, particularly bioluminescence red cell transfer, that I'm sure we're all very familiar with now, and I do a lot of work on, as does Michel Bouvier and others around the world, looking at how these heteromer complexes form and really trying to understand the relevance of them in real time in live cells and then hopefully more into physiologically relevant tissues.
Absolutely. And Kevin, if I could play off of what you said a little bit, I always found it really funny that the GPCR world was so skeptical initially about the formation of heteromers, especially when the term GPCR is G protein coupled receptors. We're already talking about a heteromeric complex. We're not talking about something that is just by itself. So I always found it a little bit like tongue-in-cheek and funny. To add to that, I think you asked a very pertinent question, which is, do you think about GPCR heteromers or homomers or any other type of oligomer really when you're designing molecules? I would actually go the other way. We published, almost now 10 years ago, that morphine, quintessentially thought to be a mu opioid, ligand, actually activates mu delta heteromers much better at greater potency, greater affinity. So our understanding of even molecules that are currently out there is actually flawed because we are not looking at heteromers as valid targets. So fundamentally, we get side effects, we get all these kind of things, and we wonder why we get it. In fact, our philosophy at Blue has literally been, we get spatial resolution and temporal resolution when we target receptor interactions. Because these receptors are interacting, as Kevin said, they're not sitting by themselves. And those interactions have meaning. And we are looking for where those interactions are happening, and is that then meaningful therapeutically? Then that is something that we can exploit. But at the outset, chemistry has to accept that and look for it. And if we actually screen for any interactome, whether it's the dopamine one, opioid one, you start seeing very interesting things happen even with the molecules that are already out there. So, and that can also explain the effect side effect profile, actually. So I think it's a very important consideration to have, in my opinion.
Absolutely. Absolutely.
So Juan, I know that you said you did dimers as well, right? So.
Yeah, absolutely. So those are great points made both by Kevin and Ajay. And I think originally with our company at GPCR Therapeutics, I think we started observing synergistic effects upon activating one of the protomers of a set of heteromer pair. And so that really, evolved into investigating those types of interactions, looking at synergistic effects between several agonist molecules and the effect of inhibiting one protomer versus the other. And developing that sort of data set really is a nice functional readout of what I think a lot of us believed was happening, but it's really difficult to get down to the to proving the exact dimerization, for example, but seeing synergistic effects from a functional assay that are very much different than the signaling event from a single protomer is really convincing and very compelling data. So those are the types of experiments that we're trying to develop and understand, and hopefully help advance the hypothesis of heteromerization. And like was mentioned previously, I think you can add the layer of complexity then of where does this occur, what cell type, what organ, and you can start seeing the wide array of signaling that GPCRs can possess, whether it's monomeric, dimeric, oligomeric, what partner that is, and so on and so forth. And I think it's just a sort of a fascinating field to be in.
So I guess
absolutely. Can I pick up on that, Annette? Sorry. I mean, I think a very good point there you made was that it's about changing the pharmacology, and we're not just interested in two proteins being close to each other or even interacting. It's about how do they change each other's pharmacology, and I think that's very critical and really is core to the definition of what a heteromer is that change in the biochemical fingerprint. Absolutely.
Yeah, Kevin, I'm planning to put that quote you said about the complex It forms, determines the pharmacology. That's going on my next T-shirt, so. But my question is, when you start the screen though, right, I need to screen a new receptor. I don't think about whether it forms a dimer and what it forms a dimer with. I just think about how am I going to screen it if I'm looking for a new ligand, right? I mean, I work on FFA2, right? I'm going to do a calcium assay. I don't think about what other receptors might be altering the pharmacology, even though they very well may be. I just think about, like identifying an agonist. Is there a better way to look at the actual question in the screen? Should I think first about who's it interacting with? Do I need to worry about that even before I start?
I mean, I can kick off if and then I'm sure Kevin and Juan have a lot of thoughts here too. But fundamentally, I also completely agree with Kevin and Juan that these complexes are actually functional units by themselves. We've seen some fascinating things. So for instance, if you put an opioid receptor, which is GI coupled, with, let's say, mGluR5, which is GQ coupled, you can actually get GQ activation by using an opioid agonist. So there's a lot of transactivation, transinactivation, allosteric effects. There's a lot going on. And I know sometimes the complexity, and we're calling them complexes, so the complexity is natural. But that can be daunting. But the flip side is understanding that and understanding on top of that the tissue distribution that could give you therapeutic advantage suddenly makes going after that target that much more meaningful. So in ways of screening, then there are various chemical strategies. You can do fragment-based design. You can do bivalent ligands. There's all kinds of strategies you can employ. But at the core of it, what is a good target? And understanding that these are functional units by themselves. It's not some additive thing that is happening. You're getting a little bit of this, a little bit of that. No, this is a completely different receptor. Many of them actually are constitutively secreted from the ER itself. So there's a lot going on. And it is complex. It's obviously difficult and not easy stuff. But the flip side is the reward is that much more. Now, and I just want to illustrate this a little bit so I think the audience can appreciate this. You can make as strong selective a mu opioid ligand as you want. A lot of different strategies people use. But the problem is in the spinal cord and the hypothalamus, you will get analgesia. Fabulous. But it's also, the opioid receptor is also in the pre-Bozinger complex in the brainstem. You'll get respiratory depression. It is there in the opioidergic neurons that are going to project into the limbic system. You're going to get addictive side effects. So understanding, therefore, the interactions and going after maybe certain tissue areas where those interactions are happening and maybe not happening in other areas suddenly gives you an entire new way to look at the landscape. It's like looking at a map in 1D versus suddenly 2D and maybe 3D. And everything changes. And that is the definition that I think we can add when we think about heteromers.
And if I can add to that, I think I'd like to take a little step back. I think in the beginning, when we started screening and looking for different ligands, we were just happy to get some response. And that you referred to getting, doing a calcium assay and getting results. And you would most likely do it the first time in hex cells. And you obviously have other GPCRs expressed in hex cells. But I think the tools we had back in the day did not allow us to investigate what other complexes with the receptors would form with endogenously expressed GPCRs, or even overexpression was always a topic, favorite topic of reviewers, especially when you were trying to publish your work and the people were saying, well, you're overexpressing, so you might be forming homodimers as well. But I think with the development of technologies like bioluminescence resonance energy transfer or other techniques where you can very nicely monitor in a time-dependent and concentration-dependent fashion what happens within the cell, it allowed us to take a step back and be able to look at specific heterodimers or homodimers or complexes in the presence of specific ligands. And I love the idea of, selectively choosing a ligand or a pair of ligands in the context of a specific tissue to get a specific response. That's very important. On the other hand, I think the time that the ligand spends with that dimer of interest is also something to consider. which could be a really interesting way of looking at it. So you might have the same dimer in different tissues, but depending on how long, or the same receptor in different tissues forming heterodimers with different GPCRs, and then the time where that same ligand sits on both of those complexes would provide the therapeutic effect, but at the same time potentially the side effects to that compound that you're looking at. So I think time is also of the essence in there. And I just noticed my camera is frozen. My apologies.
And I think that picks up an important point about the technologies you mentioned. I mean, real-time assays are so important in this, because I think a lot of what we see in terms of bias and functional selectivity, which I'm sure you'll talk to in a moment, a lot of that is kinetics and an understanding in real time. So we probably will do high throughput screening with endpoint assays, but when we're profiling and following up those screens, looking at real-time assays is so important.
So I'm actually going to ask Aarti a question, and it's because I think that it is the other side of this, in that often when I'm screening for agonists using biochemical assays, but a lot of screens are now phenotypic assays, where you identified something and you have to go back the other way and say, what does it actually target, right? And so how do you do that? Like what methods do you use when you have to go backwards?
Okay, so when it comes to soluble proteins and when it comes to membrane proteins, it's completely different. But when we know it's a multi-pass transmembrane protein, in our group, we mainly use two label-free technologies, one is called CETSA, cellular thermal shift assay, and the other one is called in-cell chemoproteomics, photocross-linking, pull-down assay. And the idea behind is that many times these multi-pass transmembrane proteins, they're very hard to purify. And then, as Kevin said, we always want to see what actually happening in the biological relevant cell line. So in that case, these methods we can use as a tag-free. So we actually use this biological relevant system for CETSA and in-cell chemoproteomics experiment. And usually these biochemical or cellular reporter assays are very robust and throughput. But these assays do not probe the target engagement under this disease-relevant setting. So this CETSA and in-cell chemoproteomics that can bridge the translational gap between screening models and disease system, which is more biological relevant system. And I can start with CETSA. So CETSA is pioneered by Palago Biosciences, and the way it works that every protein has this fixed melting point, but when it binds to any ligand, the melting point of this protein changes, and that we can use to find out how the target engagement of that protein works. And we have a paper published in Assays 2 years before, and there we use many multi-pass transmembrane proteins, including GPCR part 2 example. And there we showed that for agonists, antagonists, and even allosteric binding ligand of part 2, we could use CETSA very well. And then the other method which I was talking was in-cell chemoproteomics, photocross linking, and that worked little different than this CETSA method. So here we know the ligand, and we know the SAR around that molecule, and we can use to form affinity probe. And again, this if you know from Scripps lab, Kravat's paper, so he used this technology, he pioneered this technology. So the way it works that we make these photoaffinity probes, and then this photoaffinity probe, it forms covalent bond with the probe and then protein, and everything is into the relevant biological system, so like a cellular system, any disease relevant system. And then we can click with biotin and then do the streptavidin pull down experiments. And this is again the label-free technique. Here we have to, of course, modify the drug molecule, but again, this is a label-free technique. And we have tremendous success finding the target of we get from the phenotypic screening of those GPCR ligands by these techniques.
Yeah, I'm a big fan of phenotypic assays. I don't do them, but I think they're phenomenal in terms of the information that they can give you. With the CETSA though, are you able to do dimers as well? Because I would think, right, a dimer is going to have a different melting point than a monomer is. So maybe that's.
I think when we do it, it is very hard to know. It is dimer. So CETSA, we can do it as Western blot CETSA, which is a classical CETSA, and then the mass spec CETSA. So classical CETSA is if we have an antibody, it becomes the targeted approach. And if we don't have an antibody, then we do this mass spec CETSA, which is discovery approach. So it depends on if we can see monomer or dimer by this antibody, or if we can see monomer or dimer by mass spec.
Right. So label free. Anybody else doing label free? Doing assays that you don't have to add tags to your receptors? Anybody else doing that?
It's so interesting. I just received today a message from Dr. Hugyadi's lab from Semmelweis University from Budapest, and they just published a paper in JBC where they're using BRET, so bioluminescence resonance energy transfer, to do label-free binding assays of ligands with GPCRs. I haven't had a chance to read it, but it sounds really exciting, and they're using the Gaucher luciferase as an energy donor to actually screen for ligand binding to GPCRs, which could be a really cool paper to take a look at.
Yeah, I mean, they've done some BRET where instead of labeling the receptors, they label like downstream targets that are activated. But those are always right. Anytime you're not actually labeling, then you have issues with what you have labeled, and is it a direct interaction or not? I mean, I know why we do it, but nobody's using like impedance or surface plasmon SPR. No. All right. Yeah, I'm not either. I think it sounds great, but I don't have the instrument in my lab, so yeah, not happening. Okay, so I'm going to step back and I'm going to ask a question of Ajay and Juan, and it's because you both brought up the fact that you work in oncology with GPCRs, and I have to tell you, right, of all the areas that GPCRs spend a lot of time, oncology isn't one of them, right? So what brought you to oncology in the world of GPCRs? Because they're not well represented in that case. Ajay, you start. Oh, I can't hear you. So maybe.
Go ahead, Ajay.
There you go. Ajay.
Oh,
I think you're
Ajay,
on
we
mute.
can't hear you.
I was muted. Sorry. No, Juan, you can go ahead first, and I'll come after you.
Yeah, I think at least for you're definitely right, Annette. I think it's what seems like a sort of a new field for GPCRs, but I think at least for GPCR Therapeutics, we really did notice that we focus a lot on chemokine receptors, in particular CXCR4. And we noticed, looking through expression databases, CXCR4 really is highly overexpressed in many cancer types, exactly. And so that alone is a reason to take a look. You have, the mechanism of SDF1 as a chemoattractant. You can really see the role of that in really, promoting, for example, cells moving into the tumor microenvironment, for example, and really having the ability to avoid any chemotherapy. And so just based on the biology of CXCR4, for GPCR Therapeutics that really made sense through initial screens. We found that these are bimolecular fluorescence complementation screens to look at coupling with other proteins. We found that it really had the potential to heteromerize with quite a variety of different GPCRs. And so that really turned into looking at, expression of CXCR4 and other GPCRs as well in cancer, and interesting patterns are observed. And so that really was the motivation for pursuing that path. And we know that GPCRs are very druggable. And so the idea to look at them in another disease context really just seems like a great opportunity that I think more and more companies and labs will start researching.
Absolutely. And to play off of what you're saying, Juan, from our focus actually for oncology has been, as you know, we're talking about all receptor interactions. So we're targeting nuclear receptor heteromers. In fact, nuclear receptors have to heteromerize many times to even function. But in the GPCR context too, like a lot of GPCRs actually are expressed in immune cells, and we now understand the power of immuno-oncology, and there are ways to, we believe, to kind of just explode that field by not looking at just the glycoproteins and a few other things that people are mainly focused on right now. But I think using GPCRs in that world can really kind of expand that. So it's still pretty early stage on the GPCR side for us, but it's a really exciting phase because I think there's a lot to unlock there, for sure.
That's fantastic. Annette, can I ask a quick question? As most of you have heard, CXCR4 is one of my favorite GPCRs, and I've worked on it quite some time. And one of the issues with CXCR4 is that it only has, which is essential for so many physiological events. So in the context of CXCR4 in oncology, you mentioned that, Juan, that you realized that CXCR4 is co-expressed with other GPCRs. When you screen, and the idea of targeting CXCR4 mediated pathways in oncology, what would be, how would you modulate it? What kind of molecule would you be looking for? Knowing that you can be blocked.
Right, that's a great question. I think this gets back to some of the initial topics or discussions that we had, that we really do believe that the GPCR heteromer has this novel pharmacology that's underexplored. And so really, I think being able to pick up sort of those nuances, a specific novel pathway really is where we are focused. So you have your, agonist, antagonist, inverse agonist classes, but really, I think those are canonical ligand classes. We really consider the GPCR heteromer as being its own new signaling entity. And so therefore, really looking at, and Annette hinted at this, looking at phenotypic screens is really a great way to sort of deduce what the effect might be of this novel pharmacology, where classical canonical GPCR pathways may tell us, you really don't want to use this type of molecule. So it's really, it's a great question. It's difficult, and we're really, trying to approach it from sort of a whole cell or tissue approach, where the GPCR heteromer is really going to dictate what that pharmacology is, without, having an assumption as to what it is that the ligand is going to do.
Thank you. That's phenomenal. I think it's such a great way of evading the question. Well, not the question, but the problem of having one chemokine receptor and one ligand that does almost everything that you can think of in the body.
I guess that I have to ask, do you use HEK cells? I'm going to guess no.
No. They're useful for many, many things. And, I think we all love working with them. But to really answer the questions that we need, getting the system as close to as, disease relevant as possible is key.
So what about you, Kevin? What cell line are you using?
I use HEK cells.
I mean, it's a way to start off, right? I mean, if you just want to do a little bit of screening, a little bit of understanding of.
Absolutely.
Yeah, so for that, it works great. But I agree with pretty much everybody else here. We need to understand the phenotype and the context of it because, by the way, the other problem that I think someone else mentioned too already, hexos have their own endogenous receptors and their endogenous effector profile, which can be so different. You go to an immune cell, from there you go to a neuronal cell. The effector profile is so different too. And we now, with some of Michel Bouvier's work, so many other things, we're understanding how those effectors actually modulate the exact G-protein interaction as well. So this is complex stuff, and moving away too much from the native tissue and native cell types is dangerous because we can then get very erroneous data. We think we've solved the problem. You go into native tissue and suddenly it doesn't work at all.
Yeah, absolutely. And then our strategy is to find some novel pharmacology in a hex cell and then partner with people very rapidly who have very good animal models to actually understand the phenotype. And then we look at that. And you're right. I mean, often we see things happening in a hex cell that predicts novel pharmacology, but predicting whether that's a good thing or a bad thing can be difficult. And often it's when you go to an animal model and that really gives you that differentiator. So I completely agree. I mean, a hex cell is a starting point, but then we definitely work up to more physiologically relevant systems as quickly as possible. And to your point about receptors in hex cells, I mean, people think hex cells are empty and we put our transfecting in our receptors and that's what's there. But we're actually doing some work now with CRISPR, and we're actually adding Brett tags to receptors that are in the cell, so including CXCR4 actually. So those receptors are in a hex cell, and we can't forget that. So very important.
Absolutely.
Yeah, same with CHOS, right? I mean, the other and U2OS, the 3 lines I would say that every lab has to have.
Yeah.
Or at least we have. Maybe not everyone, but we have.
You know, but talking about phenotypic screens, right? It reminds me, like when you want to actually do it right, and you want to, let's say, take even let's say a mouse DRG neuron or something. So we started phenotypic screens with mouse DRGs because we wanted the exact ion channel profile. But then you get like 20,000 cells from one mouse. So there are complications with phenotypic screening. And I think that's why hexosources have a place to get you started and everything, because I can't imagine doing a, in silico and post that 100,000 compound screening in a mouse DRG. That would be disastrous probably to start off.
Yeah. I mean, I don't know if you have read, I mean, I was talking about this Cravat technology, Ben Cravat from Scripps Lab. So he does this phenotypic screening using a small molecule, but using the fragments. So, and he calls it the fully functionalized fragment screening. And in AstraZeneca, we use it. We have a lot of success doing it. I mean, initially building that library of, we have a library using what Bain suggested. But then we have our own database and we have our own library, and together we have almost 1,000 molecules. And so far we have identified many different targets using this phenotypic screening.
Actually, Aarti, if you don't mind me asking a follow-up question, because I think it's relevant. How do you build a larger fragment database? Because, sometimes you get a lot of conservation, right? Once you go to the fragment level. So I was just curious how you guys think about that, because I think it's very relevant.
Yeah, so we have a fragment, what we call fragment-based drug discovery department. So they have their own fragment library. And when we do this phenotypic screening, we always do in 3 different doses. So 10 micromole, maybe 50 and 100. So sometimes 100 micromole is too much. But then whatever and initially we get some hits. For example, out of those 1,000 molecules, we get 20, 30 molecules as a phenotypic hit. But then when we do the dose response, not all pan out. So whatever hits we get, from finally using this dose response, we do these target deconvolution efforts using in-cell chemoproteomics, which I was talking about. And then, so here the main aim of our phenotypic screening is to find the target, so not to find the ligand. So once we always want to have a new novel target. So that's how we do.
Gotcha. Thank you.
I think it's such an interesting way to screen, right? Sort of backwards, right?
Yes.
It's almost, so I've always loved pharmacogenomics because I think of it as being unbiased, right? I mean, I love the genetics part of it, right? When you look at, Juan, as you mentioned, right, that you went back to the informational databases and found that CXCR4 was just upregulated all the time in cancers, right? That's unbiased information. And I think sometimes when you use a phenotypic assay and you go backwards and look for the ligand, it's very unbiased. I don't have a target that I love that I need to find a ligand for. I go at it by, I have something, a problem I need to solve, in essence, that, you know, whatever solves it, solves it. But the problem has often been when you go backwards that you identify potentially a target that you're really not interested in, right? I mean, that you don't really want to turn off maybe or alter, I don't know. At least I would think that would be occasionally a problem. All right, so I want to ask a quick question about structures. We have to talk about structures, right? We're working on GPCRs. The evolution of crystal structures in the world of GPCRs has certainly changed over the last 10 years. I mean, while we don't have an image for everything, right? I don't have a crystal for every GPCR. I've certainly got a lot more than I had. Do you use that information when you're screening? Do you use any kind of rational drug design in your approach when you're thinking about targeting a receptor? I'll start with Juan.
Yeah, so like you mentioned, you know, the advancements in the field are just amazing. It seems like, you know, every month there's new structure or several coming out. So it's just a lot of fantastic work. And I think if it isn't already, I think most GPCR drug discovery programs will be utilizing that information one way or another, whether it's through virtual high throughput screening, if your target's available. I think it's a phenomenal way to really, you know, cut down on potential, you know, non-hits and really save resources on the wet lab side. So I think it's really, all that information is super valuable. Being a heteromer specific company, we really would, you know, like to see the heteromer crystal structure, which I think is still, there's definitely dimer structures. CXCR4 itself was solved as a dimer. So understanding the structural information of a heteromer would be supremely valuable for a company that's focused on dimers. But I don't think the field is there just yet. I think there's complexities in isolating the, you know, for example, a heteromer pair. So I think for in the general GPCR drug discovery field, using structural information, I think it's going to become common practice if it isn't already. I see it in the near future as really being part of a foundational type of program for any company.
Yeah, I can tag on to that. You know, completely agree with that assessment. And I still remember in 2007 when the Kawatkar lab published the adrenergic receptor crystal, and it was suddenly like, wow, like this is actually possible, and it's not all just based on rhodopsin. And you know, in the heteromer context too, there's good sense now of the conservation of interfaces where heteromerization is happening, or oligomerization, just to be fair. And I think that is also very important, right? Because many times we think about, you know, oh, they're just two things, they're stuck together, but then the orientation of how they are stuck together matters a lot. And how you can then bridge that. Is the allosteric site on the other side available? And so many design factors So we take all that into consideration. There's, the in silico portion can cut down. I think Juan talked about this too. Like you can spend a year, trying to figure out how to work with it. And if you have good structures, which fortunately we do, we don't have a heteromer structure, but at least we understand the interfaces where heteromerization happens, so that it's already helping us immensely. We couldn't have done this probably 15 years ago, to be honest. It's so much harder.
Absolutely.
So Kevin, does it change the way that you screen at all?
Not necessarily the way we screen, but certainly thinking about interfaces. We're starting to see a lot of really interesting transactivation, has been mentioned before. Not necessarily just between GPCRs actually, but between other receptors and GPCRs and kinases, absolutely, and other receptor molecules. So it's interesting to understand those interfaces, because I think those become a really interesting therapeutic target. And we're always looking for new targets. And actually, I think there's still an awful lot of target discovery out there where we're actually looking at a heteromer complex as a novel target.
Well, because I'll just follow up on that, right? Because the advantage oftentimes by having a target of a protein that's associated with the GPCR is that it may provide you with tissue specificity, for example, right? I mean, isn't it really the idea that you have a means by which to make it a more targeted drug?
Definitely. And I think that's one of the great selling points of these complexes is the tissue specificity, and that's been seen on a number of occasions. And I think the targeting of that, and therefore reducing the side effects. And that's where a lot of this is being driven from, is the reduction of the side effect profile. So
we have to come back to it, because of course I love functional selectivity, right? I'm all about the G protein. Do you really, I mean, when I originally thought compounds that could induce coupling between a receptor and a given G protein, I mean, there really wasn't even language for it back then. We made it all up as we went. But I think now it's clear, right? I mean, you have receptors that couple to more than one G protein, that couple to arrestin, that you can really determine sort of the downstream effectors based on your ligand that you start with, right? I mean, you have chemokine receptors that just the endogenous ligands for the chemokine receptors have a multitude of G proteins that get activated downstream depending on which ligand you give them. So I think, do you think of that as being an issue then when you think of like deconvolution? I mean, it's hard enough when you just think of one simple receptor, one G protein interaction that you're having to identify. What happens when you have to figure it out among, such a broad array?
I think one of the challenges is understanding which particular signaling pathway has the therapeutic effect. So I think functional selectivity is an amazing. I mean, it's like heteromerization. It opens a Pandora's box, and it becomes incredibly complicated incredibly quickly. And drilling down to which pathways are really causing the effect. I mean, for example, in the vasopressin system, I mean, it generally looks like it's all about one G protein. So, I mean, there might be other G protein interactions, but as far as we see, it's about the one G protein pathway. So, what deconvolution I think is the key to this, and coming back to the HEC293 cell, if it happens in a HEC293 cell, does it happen in the body where in the target organ? So that's the challenge, I think, is deconvolution.
And if I could just kind of add a point to that, and this is something that I wonder, and maybe someone here knows the answer. Is there also such a binary aspect to it? Can a ligand, for instance, be just biased towards one effector? Could there be, for instance, temporal nature to it? For instance, it could be G protein activated in certain contexts it could be beta-arrestin, it could be some other effector. And could that be happening even in the same tissue, but based on different temporal or other context, disease state context? So these are some of the questions that like boggles the mind for me. I don't know if any one of you has any knowledge in that. I would love to hear about it.
Well, they know that, right? For like beta-2, as beta-2 becomes adrenergic receptors, you become more phosphorylated. You switch from GS to GI, right? So it's a phosphorylation mechanism. And there's all kinds of papers about the barcode, in essence, of the GPCR, right? And it helps identify what G proteins are going to interact, or what arrestin is going to interact with the receptor based on the phosphorylation status of the receptor. I think a lot of ligands activate more than one G protein, and in fact, both the G protein and the arrestin. I mean, but I think, right, what we want is something like, can you make it selective, right? I mean, can you make it just GI coupled or just GS coupled, and ignore all the other stuff that would otherwise be activated? I'll tell you, we recently did a screen on a receptor, and we thought what we wanted was GQ coupling, but it turned out as long as we turned off the beta arrestin, we would push enough down the GQ, we were fine. So we could look for just antagonists of beta arrestin or agonists of the GQ coupling. You know, you didn't have to pick agonists of GQ. You could just turn off the other and you were okay. So, you know, I think when you think of functional selectivity, it may just be, you sort of need to fine tune, you know, which G protein you're turning on downstream. But to your point, I don't think we always know, right? I mean, I'd be willing to bet the vast majority of receptors, they have no idea what coupling to G12 or G13 does to them. We talk about GI, but what about GO and GZ? You know, with the G proteins that are harder to turn off and turn on selectively, it becomes harder to know physiologically what's the relevant in a cell, right? You mentioned cancer, right? I mean, a lot of immune cells, right, they have G16. They don't even have as much Q11 and 14, right? They've pushed to a different pathway. So it doesn't matter. And for me, I actually think about things like, what about age? What about the age of the cells? Does it matter? Because your pattern probably changes, right, as you age in your cells for what G protein you express. So I'm going to have one more question, and it's because I can't not talk about it, even though it wasn't on my list of questions. Somebody mentioned nuclear receptors, and of course that immediately made me think of, do you look at where your receptor is in the cell? Do you look at it intracellular? Does it matter? Do you care? When you're looking at your GPCR, because it's a hot topic right now, right? In terms of receptors that are not necessarily at the plasma membrane surface. So, do you look?
We certainly do. I mean, we've spent a lot of time recently looking at intracellular trafficking and developing markers to look at where in the cell the bread is happening, and following that in real time.
You're doing it with the bread. You're like looking at the location based on the complementary bread proteins. Okay.
Yeah, absolutely. So using the Rab proteins. So this is leveraging the work from Neven Lambert and using a system where there are Rab proteins in different compartments and using that to follow the receptor around the cell. And we've developed that a little bit further, and particularly looking at forward trafficking as well as a novel approach to look at where that functional effect is coming from the heteromer complex.
That's awesome. And they're inside the cell, right? Your receptors?
Yeah, absolutely. I mean, obviously they internalize, and a lot of heteromerization is around the forward trafficking and how these are chaperoning each other up to to the surface. So there's an awful lot of pharmacology. And David Calabaro over in Birmingham is doing some amazing work looking at the intracellular domains where you've actually got actin cytoskeletal domains that are sort of ring fencing this activity. So you can monitor these with single molecule techniques to actually see these receptors very much compartmentalized within these little domains in the cell. So I think again, cells are not just bags of water. They're very complicated things, and yeah, actually understanding where this is happening is I think the next domain of endeavor for our field.
I actually completely agree with that. But when we were working with, for instance, DRG neurons, if you think about physiologically, the ganglion is by the spinal cord, but the nerve endings are peripheral. If we think about what is happening in that context, it's like something is happening in Boston and the cell body is in Seattle, and so how is this signaling even functionally, relevant, right, in that context? So we've been thinking about these things, signaling endosomes. Is there constant level of tone that is maintained? So these are all great questions. I don't think we are there yet to answer a lot of these things, but I think we're plugging away at it. Understanding that, I think, is therapeutically relevant, because otherwise then we're treating the wrong problem in the wrong place, or the right problem in the wrong place. Either way, we're in the wrong,
yeah.
Exactly.
Anybody else?
Absolutely. Yeah, just agreeing with what's been said. I think it's something that's, of utmost importance. And I think there's been enough studies to show that a lot is going on in different compartments within the cell. And I think it's, like AJ mentioned, I'm not sure that we're completely there yet, but being aware that some of the effects that are being observed could be due to endosomal signaling, for example. But, we're kind of just scratching the surface, and, literally first step, make sure that your receptor that you're trying to target from a drug discovery standpoint is at the membrane surface, and then going from there. But really building in all those other layers, I think, is definitely a next step.
I love it. I can only echo what's been said. I think it's important to know who, where, and when these things are happening, and are these physiologically or pathophysiologically relevant in order to specifically target these with molecules that we'd screen for.
Okay, so with that, I'll have my final question, and it's not your if you were a drug, what would you be question, but it's close. So where do you see GPCR screening in 2 years? Like in the future, what would you love to have, kind of thing? If you were Harry Potter and you could come up with your own screen for a GPCR, what would it be? What problem would you solve?
I'd like to see some structures with dimer interfaces, and that was picked up earlier. So that's what I'd like to see.
Yeah. Yeah, I think dimer or heteromer, maybe cryo-EM complexes with signaling partners, whether it's beta-arrestin or G protein, or maybe multiple G proteins. That would be fascinating.
Yeah, I to tag on to that structure is, I think, the very key area. Secondarily, to add to it, I think the microscopy world too. Like, can we get better resolution? Can we find ways to actually get native tissue, determination for heteromers? Right now we use PLA. There's very few techniques that are out there. If there's any development on that, that would be fantastic.
Yeah.
And I think I'm going to take it a little bit out of space here. What I'd love to see is have bioinformatic tools, AI, machine learning tools to help first of all, what I would like to see is a database where all of this information that's available now is compiled, organized, and then have the tools to explore these questions in a dry lab before even stepping into a wet lab and doing any experiments. And having that ability to kind of, I just rewatched the movie The Matrix, and having that ability to see the code before even stepping into the lab, I think would be really useful. It would decrease spending when it comes to, you know, doing experiments in the lab, but also it would save a lot of time and hopefully advance the field, which is the goal for everyone.
Yep, I agree to that. I mean, now with this AI learning, we have so many dry lab stuff we can do it. So if we can expand on that, it will be beneficial to everybody.
I'll tell you that mine is, of course, pharmacogenomics. I want to know what all the natural variants that people carry with them in their GPCRs, how that affects their response to drugs. And I think there should eventually be a database for that too, but there's not much out there now. So maybe in a couple of years. So, all right. Well, I have to thank each and every one of you on the panel for joining us today, and I thank the audience for attending.
Thank you.
Thanks.
Thank you.
Thanks for having us.